Good morning. It's June 24th, and this is your daily brief in science. Here's everything you need to know. A recent study from the University of Edinburgh has unveiled a remarkable breakthrough. Genetically modified E. coli bacteria can convert plastic waste into paracetamol, a widely used painkiller. This research specifically targets polyethylene terephthalate, or PET, a common plastic that generates over 350 million tons annually and is notoriously difficult to recycle, often ending up in landfills or oceans. The innovative approach combines traditional chemistry with engineered biology, potentially fostering a circular economy and reducing reliance on fossil fuels. Published in Nature Chemistry *, the study highlights a unique chemical reaction known as Lawson rearrangement, which can now occur safely within living cells, a process previously only observed in laboratory settings. The research demonstrated that this rearrangement, typically requiring harsh conditions, can spontaneously occur in E. coli with the aid of phosphate, showcasing the bacteria's potential for complex chemical transformations. The conversion process achieves an efficiency of up to 92% in under 24 hours, indicating its practical application for pharmaceutical manufacturing. Current production methods for paracetamol heavily depend on fossil fuels, starting with phenol derived from crude oil, contributing to climate change and environmental damage. This new method could significantly reduce the pharmaceutical industry's dependence on oil-derived medications, promoting a cleaner approach to drug production. Future efforts will focus on scaling this process for industrial applications and conducting life cycle assessments to evaluate its environmental impacts. However, scaling this method for industrial use presents challenges, as noted by chemist Dylan DeMaul from the Colorado School of Mines. Collaboration with companies like AstraZeneca is already underway to further develop these innovations in sustainable chemicals and materials, emphasizing the importance of sustainable healthcare practices and initiatives aimed at reducing plastic waste. A recent study sheds light on the role of newly evolved genes in regulating the cell cycle, a critical process for cell division. This research takes an interdisciplinary approach, merging cell cycle biology with genomics, which allows for collaborative hypothesis testing and experimental design. The significance of analyzing gene activity changes during cell division is underscored in this study, showcasing the collaborative nature of the work. Key contributions were made by researchers Romain Faure, Cyril Pulver, and Alex Lederer, who developed a detailed atlas of gene activity throughout the human cell cycle, making this information accessible to both researchers and the general public. Among the notable findings was the identification of ZNF519, a gene that is unique to primates and essential for accurate DNA replication before cell division. Disruption of this gene results in slowed cell growth, highlighting its importance in the cell cycle. Additionally, the study revealed that knocking down specific transcription factors can cause cells to become stuck at various stages of the cell cycle, disrupting normal division processes. Another key protein identified, ZNF274, is present in mammals but absent in older species, and it plays a role in regulating the timing of genome duplication, which affects epigenome maintenance and nuclear organization. The researchers express optimism that their comprehensive resource on human cell cycle gene expression will support scientists globally in advancing studies of cell biology and disease. These findings may also provide insights into the differences in cancer prevalence and behavior between humans and other mammals, contributing to a broader understanding of developmental disorders. A research team from the Institute of Science Tokyo has achieved a significant breakthrough in remote-controlled microflow using light-responsive DNA condensates. Led by Professor Masahiro Takinue and Assistant Professor Hirotake Udono, their findings were published in Nature Communications on May 14, 2025. This study details a novel method for producing mechanical actions in DNA structures, an accomplishment not previously seen in similar research. The innovative approach involves the use of photoresponsive DNA liquid condensates featuring azobenzene. This allows DNA motifs to dissociate or reassemble in response to ultraviolet and visible light. Experiments conducted by the team demonstrated various mechanical action modes, including the outward spread of condensates under UV light and a gel-to-liquid state transition. Notably, the DNA condensates exhibited unique directional motions, resembling jellyfish swimming at lower light switching frequencies and displaying a Pac-Man-like movement at higher frequencies. This indicates the potential for robotic actuation through these structures. To enable these mechanical actions, a non-photoresponsive DNA motif was cross-linked to the photoresponsive structure, facilitating the generation of microflows. The implications of this research are substantial, paving the way for advanced applications in fluid-based diagnostic chips and molecular computing. The researchers created Y-shaped nanostructures by self-assembling single-stranded DNA strands with azobenzene integrated into their terminal ends, enhancing their photoresponsiveness. By alternating UV and visible light, they demonstrated a spread and collect mode, showcasing the condensate's ability to undergo reversible phase transitions. This study marks a pivotal advancement in the field, introducing a method for achieving mechanical actions in DNA structures that had previously eluded researchers. A groundbreaking study published in Nature Cancer reveals how inflammation triggered by cancer-associated mutations can reprogram mutated cells, setting the stage for tumor initiation. Led by Professor Cédric Blancpain from the Université Libre de Bruxelles, the research highlights the importance of understanding the molecular mechanisms behind this cellular reprogramming. The findings suggest that targeting inflammation could pave the way for new therapeutic strategies aimed at preventing prostate cancer and other malignancies. Notably, the study indicates that pharmacologically blocking inflammation can halt cellular reprogramming and prevent the initiation of prostate tumors. Prostate cancer is a pressing health issue, being the second leading cause of cancer-related deaths among men globally. In Belgium, over 12,000 new cases are diagnosed each year, underscoring the need for early detection to improve treatment outcomes. The aggressive nature of prostate cancer is underscored by the recent disclosure from former U.S. President Joe Biden, who revealed his diagnosis of an aggressive form of the disease with bone metastases. The study identifies two types of cells in the prostate, basal and luminal, that can initiate cancer in different regions of the gland. Dr. Jiang, a key researcher in the study, noted that the markers identified could serve as predictive biomarkers for aggressive prostate cancer. The research also established a correlation between inflammatory activation and severe prostate cancer cases, linking findings from mouse models to aggressive cancers in men. This significant collaboration between the Université Libre de Bruxelles and the Katholieke Universiteit Leuven, supported by organizations such as FNRS and the European Research Council, represents a vital step forward in understanding prostate cancer. Researchers at Oregon Health and Science University have made a significant discovery regarding the structure of glutamate receptors in the cerebellum. This breakthrough could lead to new therapies aimed at repairing brain function affected by injury or genetic mutations. Using advanced cryo-electron microscopy, the team mapped the molecular structure of these receptors at near-atomic scale. This mapping revealed critical insights into neuronal communication that impacts movement, balance, and cognition. The study highlights the precise spatial arrangement of glutamate receptors, which is essential for effective synaptic communication and neurotransmitter signaling between neurons. Senior author Eric Guo emphasized that understanding the molecular structure of these synapses is crucial for developing potential therapeutic interventions to restore cerebellar function. Co-author Lawrence Trussell noted that injuries or genetic mutations in the cerebellum can lead to severe disorders affecting balance, movement, and cognition. The findings suggest a promising direction for developing drugs that target these glutamate receptors to enhance cerebellar function, with implications for treating various neurological disorders. Although immediate treatments are not anticipated, this foundational discovery may inform future therapies aimed at restoring function in cases of injury or genetic disruption. The research, published in the journal Nature, was conducted by a team at OHSU, including lead author Chengli Fang, and supported by the National Institutes of Health and the Howard Hughes Medical Institute. This study underscores the importance of properly organized synapses for efficient neurotransmitter signaling. Disruptions caused by injuries or genetic conditions can lead to motor and cognitive disorders. Overall, this research could pave the way for new therapies aimed at repairing disrupted neural structures affecting motor skills and cognition. This has been your Daily Brief in Science. To read more about these stories, follow the links in the episode bio. You can also subscribe to these updates via email at www.brief.news. For more daily podcasts about the topics you love, visit brief.news/podcasts. Tune in tomorrow. We'll be back with everything you need to know.